Morning, sir.
Actually, I'm really sorry because I actually messaged you in Telegram at around 7 o'clock, but the mobile internet was actually off, so message was not sent.
It's okay, sir. No
Later, when I
problem.
got a call from Mayuga, that's when I knew that, oh, yeah, shit, I forgot to on my internet. Okay, okay, so let's begin the class now. Okay. So today we're going to discuss the basic of this. We're going to start with this structural proteomics part. So before I begin with the structural proteomics, let's discuss a little bit about how the entire proteomics workflow actually. Okay. So first, what as I've told you that what's the major difference between proteome? What is proteomics? The proteomics is actually a study of the entire proteome because as generally happens is that we don't study the entire proteome, rather we study a single protein, but every protein actually works by interacting with other protein, and therefore fluctuation in one protein leads to the fluctuation or activation or inhibition of the other proteins. And this is not about only two proteins, there are multiple proteins in a single signal cascade, right? Whenever we talk about signaling, whenever we talk about something, every thing in our body is controlled by this signaling cascade. So, be it apoptosis, be it angiogenesis, be it cellular growth, be it cellular differentiation, be it immunology, whatever you talk about a particular domain that is controlled by a variety of proteins that works in tandem with one another, and that is known as the signaling cascade. Okay. So generally the proteomics is actually divided into three parts: the expression proteomics, the structural proteomics, and functional proteomics. The expression proteomics, as the name suggests, depends upon the, or it works, or it actually covers the domain about the expression of a particular proteome. That means expression of a particular protein, how much that particular protein is being expressed, and what tissue is it being expressed. These are the things that is covered by the expression proteomics. Okay. But in everything, what you have to do, you have to collect the sample. The sample is actually the pre-fractionation. The sample might be a tissue culture, it might be from the cellular extract, it can be a tissue extract, it can be an organelle extract. The system might vary, but you have to collect the entire thing and then you have to process it, because once we collect it, all the proteomes are inbuilt into it, right? Whenever, suppose I am collecting the tissue sample. So when I'm collecting tissue sample, I'm getting every protein that are present in the tissue sample. So we have to separate out the proteins, because if we don't separate out the proteins, it's really difficult. It's going to be very difficult for us to understand which protein is getting overexpressed or which protein is getting inhibited. So for that, we generally use the gel-based proteomics, or we either under 2D gel electrophoresis or the 3D gel electrophoresis. Okay. Now that actually separates the protein, and once the protein gets separated, then what? Then we have to perform the mass spectroscopy to identify the particular protein. So generally, we perform MALDI-TOF, BSI-TOF, or LCMS-MS depending upon the type of experiment we want to do, or depending upon the answers that we want. Okay. And on doing this and performing that, we can identify the proteins that are present in my sample. Then we use the data analysis to understand what, because you know in MALDI and in LCMS, what happens, there are vaccination of the proteins. So the proteins actually get vaccination if you use the SIMS. So then you have to rebuild the entire protein structure, and that is the data analysis. For that, we have to take the help of several databases. And once the databases like Sequus, Mascot, Comet, Tandem, Denovo, these are all databases. You take the help from those databases, you build up the entire structure, and then we can get to know about how or which protein is getting affected or which protein is happening or that. Okay. So, but as the name suggests, this structural, we're going to focus on the structural proteomics part, specifically on the modeling part, the homology model. Okay. So, as the name suggests, the structural proteomics give information only not about the expression of a particular protein, but about how the, what is the role of that particular protein, and what is the SAR. SAR means structure activity relationship. how does the activity of one structure is controlled by activity of one protein is controlled by the structure of the particular protein, or vice versa. How does a particular structure can manipulate a particular activity inside the body. So this is structure proteomics. Okay. Now generally what happens is that first we only have the protein 2D structure. So what do you mean by 2D structure? How many levels of protein structures are there?
So generally, we have primary, secondary, and tertiary structure.
Very good. And what is primary structure?
So basically, in primary structure, we have a single chain peptides, and
Single chain peptide. Okay.
sorry if I'm wrong because I have studied in third semester. I almost forgot. But in secondary structure, generally. The secondary structure, if I'm not wrong, there are more than one chain, and in the tertiary structure, the coiling of the secondary structure, like into certain structures like beta hairpin and all, that constitute of the
Okay, we'll
tertiary
see.
structure.
We'll see about that. But generally, what happens sometimes the protein is made up of one chain. So if protein is made up of one chain, then in either case, beta, secondary, tertiary, or primary, you're going to get a single chain. Okay. But why is this importance of structural proteomics is there? Importance is there because it is very difficult to structure a particular protein using the standard method that we have. And what are the standard method of protein structure prediction?
So, can you repeat your question?
Which experiments are usually done to for protein structuring?
So generally, I think just like spectrometry, we have certain structure predicting. You know,
Generally, we
also
use
in chromatography, we have certain.
generally in chromatography you don't get structure. Okay, we can purify the protein through that. But yes, it's right. We use spectroscopy, but what spectroscopy? We use NMR spectroscopy. Okay. Generally,
In
there are three methods for protein structuring. One is NMR spectroscopy, the X-ray crystallography, and the cryo-EM. Okay, the cryo-electron microscopy. So these three are the method which can help you to predict the structure of a protein or a DNA. Okay, any structure of any macromolecule. But sometimes these are not enough because for structure prediction using these three methods, you have to have a protein in high concentration, or you have to purify the protein, or even if you are able to purify the protein, you don't get enough amount of protein. Okay, so these are the issues, and generally there are certain proteins like membrane protein which are really difficult to extract. So these challenges or these shortcomings of this actually prevent us to perform or predict the structure of every macromolecule. So that is why structure proteomics part has developed, so that we can use the bioinformatic power and the power of computation to predict the structure of a particular protein. Now, as you know that for protein structure, these are generally there are 20 amino acids that are there, and these 20 amino acids make a structure of a protein or the random management of this particular amino acids. And each amino acid have their own specific chemical and physical properties. For example, you see these are the, if you check about these residues, these are the positive charged molecule, whereas these are the negative charged amino acids. Some molecules or some amino acids are there which have polar and charged side chain, and again you have the molecule with hydrophobic side chains. Now obviously the molecule which are polar side chain, they can interact with water, they are soluble in water, whereas the hydrophobic side chain, as they are hydrophobic, obviously they are not going to mix well with the water. But in our body, the we have 70% water, right? So
Yes, sir.
whether we have a hydrophobic side chain or a hydrophilic side chain, doesn't matter. The protein is soluble in water. But how do you make something which is not soluble in water soluble in water? So that is the challenge, and that is the thing that we have to understand. So Now coming to the protein structure, as you know that there are four different structure level for a particular protein. Four different. What? First is the primary structure. Now, what is primary structure? Primary structure is the sequence of the protein, starting from the N terminal ending to the C terminal. That is the sequence of the protein. So it starts with a single amino acid. So amino acid is simple, the amino acid chain. Amino acid 1 connected to amino acid 2 connected to amino acid 3 connected to amino acid 4. This is the primary structure. Here we only have the sequence of the protein. In secondary structure, these amino acids starts to interact with one another. They start to complement each other. For example, as you can see in this particular case, see here what happens, this particular carbon, the CO is making, or this particular NH is making a hydrogen bond with this particular CO. Now this is actually amino acid 1, then you have amino acid 2, and then you are going to find the amino acid 3. So this molecule is actually making a connection with amino acid 4 over here. As you can see, this is 1 2 3 4. So this is I to I plus 4 connection. Similarly, like that, this, what is this kind of a, this is a hydrogen bond. Okay, now like this, several hydrogen bonds starts between the amino acid. As you can see, the third amino acid is making a hydrogen bond with either the fifth or sixth amino acid. So this kind of I to I plus 3 or I to I plus 4 interaction, I is the ith amino acid and I plus 3 is the I plus 3th amino acid with which it is forming hydrogen bond. Now generally there are two kind of secondary structure. One is alpha helix, another is beta sheet. And these are differed by the way hydrogen bond perform. These are differed by ways the structures, this hydrogen bond leads to a particular structure. Okay. So generally I to I plus 3 and I to I plus 4 leads to alpha helix structure, wherein it's simply like a coil. It's simply like a coil spring. Each is coming in close contact with the other. Okay. In case of beta sheet, we can find out there are hydrogen bonds, but the hydrogen bond is not between I to I plus 3, it is between two chains. This is the intramolecular. See one chain of beta sheet, another chain of beta sheet, they are connected to one another. So therefore, this is why two beta sheets are always in close contact with one another, and for beta sheet you generally get I to I plus 5 connections. Okay. But the thing is that these are part of the chain. Now, not the entire protein is not going to fold into an alpha helix, or the entire protein is not going to fold into a beta sheet. As you can take a look from here, this is a small fragment. This is a particular secondary structure of a small domain. This is another domain. This is third, fourth, another domain. This is the beta sheet domain. This is a varied domain. So you might have a very long amino acid. The amino acid sequence, there might be 500 amino acid in a particular protein, but it is not forming a single structure. There are mixture of three kind of structure. One is alpha helix, another is beta sheet, and the third one is the random coil structure, which does not have any proper structure. We generally call it random coil or loops. Okay. Now what happens is that this particular secondary structure again fold into one another and interact with one another. They fold over one another and make a complex structure. This is known as the tertiary structure, where the entire protein gets a particular conformation. Okay. So this is about a single chain. A single chain, it folds to a particular structure. That's the tertiary structure. But sometimes proteins are not made up of single chain. They are made up of multiple chain. For example, the hemoglobin. How many chains do hemoglobin have? Monishika?
Yes, sir. Yes, sir.
How many
Yes.
chains do hemoglobin have?
So generally two, if I'm not wrong.
Not two, four. Alpha 2, beta 2. Right, there are two alpha chain, there are two beta chain. So this is a structure of the hemoglobin. Okay, so there are two alpha, two beta. So when several of this molecule more than one chain of the molecule comes together, interact with one another, or several tertiary structures. Four tertiary structures in this particular case of hemoglobin. There are four tertiary structures that come in contact with one another and they form a particular structure, and that is the quaternary structure. Okay.
Sir, I have a question over
Yeah.
here
Yeah.
because just a second. I'm trying to zoom the screen. So actually, when you said here I2, I3, and I5 kind of bonding, because you said the amino acids are bonding with each other
Each
in
other.
the alpha helix.
Yes.
So what do that I signifies? Matlab, it is signifying the amino acids.
ith amino acid, or you can say n to n plus 3, anything.
Okay,
I
sir.
signifies a random number.
Okay, sir.
Okay. And I plus 3 means 3 amino acid away from the I.
Okay.
Okay. Thing is that, so once we model this, once we get this, then we have a proper protein structure. Okay. As you can see, but the thing is that once we have a protein structure, or once we model a protein structure, the protein structure is very complicated process. It's a very complicated process. Now, how do you know that a particular protein has taken a proper folding? The protein has properly folded. How do you know that? If I simply say that you have this hemoglobin, Konishika, take this hemoglobin. Let me know if the protein is properly folded or not. So, how do you know?
Sir, like we have to perform some tests. Like as I said in the previously, I have said about this. So you said we cannot perform chromatography for specifically the size, but sir, can't we
No, we
perform
can perform chromatography. I said that chromatography
like
does
the
not give
size?
you a structure. It is used
Yes,
to purify the protein.
yes, sir. But so when we are performing the column chromatography, which is the size illusion chromatography,
Yeah.
so can't we get the structure? Like as you asked a question that how do we know the proper folding has occurs? Sir, it also gives us the idea about the foldings of the protein.
Yes, it gives you the idea about the structure.
Yes, sir.
Is it a globular protein? Is it unfolded? Only that, nothing else. For
Okay.
example, if I simply ask you that how does a person look like, or how does your father look like? If you simply say that yes, he has two hands, two legs, two eyes, and a head, so that is true.
Okay.
What you are stating is obviously true, but that does not allow me to understand your father's facial characteristics, like right?
Okay,
So
sir.
that is chromatography. Chromatography allows you to separate person. Through chromatography knows, and chromatography can tell you that yes, they have two hands, there are two legs. But how would I know if the hands are longer or shorter?
So
How would I
then,
know the height of the channel? How would I know that the exact nature? So for that I have to take a photograph, right?
yes, sir.
So protein structuring is like taking a photograph. We all know how the protein generally looks like. Either it is globular or it has a long stretch. We know that because it is made up of only 20 amino acids. But what exactly is the structure? For that we have to either make a portrait or we have to take a photograph. Correct?
Yes, sir. So,
Okay.
sir, in that case, we can do like spectrometry.
Spectrometry to get protein folding. Very good. But how do you conclude?
So, generally, what will happen? Basically, we use NMR or UV light. These are the common spectrometries. So, or extra crystallography. So, generally, how the light is been absorbed or how much is deflected give us a kind of picturization of how the protein is been folded. So, we can get the idea from there.
But through NMR, NMR is for protein structuring. You are going to get atomic detail about that. That's good. But if I simply give you a protein right now, it is going to take you around 1 month to solve the structure. Only by solving the structure you can say it is properly folded or not. That's a too complicated process. I'm simply asking just by anything which allows you to give you whether the protein is properly folded or not. Just in a second. Within 10 minutes you're going to know this protein is either folded or not. ponder
No.
over that question. I'm going to answer that later. So there are some experimental techniques too, but the best way to understand if the protein is properly folded or not is to analyze its Ramachandran plot. Okay. So now, what is Ramachandran plot?
So no idea.
Okay, very good. So this picture over here denotes the Ramachandran plot. You see, in this particular protein structure, what happens? There are several bonds. The first is the C2 C alpha bond. The C2 C alpha bond is known as omega bond. Then you have, there are three bonds: phi, psi, omega. Okay, sorry. The C2 C alpha is phi. Okay, the N to C alpha is psi, and then we have N and C, which is omega. Now, the major importance about this particular bonding is that though they are free to rotate 360-degree, practically doesn't happen so there was a scientist G.N. Ramachandran who actually deduced and measured the phi and psi angle of the proteins, and he found out that though theoretically this phi angle, phi psi have the tendency to have any angle, any desired angle they want, but practically it is not always so. They always have some restriction and they try to gather in some particular area. So when he mapped the phi psi angles of a lot of protein, he found out that in this entire chart, the red zones are the allowed regions where a particular protein stays. The phi psi angle of all the amino acid of that particular protein tend to cluster to this particular allowed regions, whereas the gray zone is known as disallowed regions. Okay, so see, there are a variety of regions for a particular alpha helix. Generally, the phi psi angles correspond to this exact location, to this particular location. Whether for a 310 helix, generally the phi psi angles are located over here. For left-handed alpha helix, it is present over here. For the beta sheets, parallel beta sheet, as you can see the names, they are conserved over here. Okay. So though the protein is free to have any value they want, generally they don't have that one. But so now if you make a Ramachandran plot for a particular protein and you see, yes, there are structures, there are phi psi angles in this particular location, there are phi psi angles in these allowed regions, then you know that your protein is not properly folded, because a properly folded protein has clustered phi psi angles only to the allowed regions. Is this part clear?
Yes, sir.
Okay, very good. So now there is again one question: why this clustering is done? I mean, what do you think? That though they are free to have any angle they want, why don't they have one? Why are they clustering to only a few sections, not everywhere on the screen?
So I feel they're clustering into specific regions because they can make some intra bonds between the groups.
Good. The thing is that see, when the proteins, you are actually right, because though they can have any bond, for example, for this alpha helix, see, the pi psi bond can have any angle they want, but since they are connected by this hydrogen bond, they have a restricted movement, right? They have a restricted movement. Again, now you might say that for the loops, this loop region, I understand that for beta sheet and alpha helix there is a restriction, but for these loops there is no restriction, right? So the loops might have any angle they want.
Yes, sir. So are you showing something on the screen because the picture is quite zoomed out? So can you
I'm
please
only
repeat
showing a
your
random loop. Okay, so
Okay, okay.
let me show you just a minute. Suppose see, in this particular case, this is the alpha helix, right? This is the alpha helix part, right?
Yes, sir.
This is the alpha helix. This is the beta sheet. Whereas these are the loops. So I understand that this alpha helix and beta sheet, they are connected by a particular bond. So they are restricted movement. But for these loops, these are the loops region. Okay, these are the loops. They have random structure. Loops have no structure at all. At that particular moment, they had this structure, but again, at the next section, they're going to have a different structure. So there, they have a different orientation. They don't have a particular orientation, but they also obey this particular rule of getting congested to only a small section.
Yes, sir.
So why do you think that happens? I don't
Sir,
care if you're right or wrong. Simply tell me.
sir, I feel uh, maybe because of some other forces like mm, the hydrophobic interactions or maybe like static hindrance, something like that.
Not exactly that. See, the thing is that though the loops are free to move, they are free to move anywhere they want, but at the end they are actually connected, right? The two terminals are connected.
Yes, sir.
If I simply talk about this particular loop, these two particular loops, the terminals are connected. They are not free. So even if they are able to move to a particular orientation, that particular orientation which is not going to happen. For example, suppose you have a long stick. Suppose let's take a look about this particular option. Suppose you have a protein. It's a long stick. Okay. So even if this particular entire atom moves a lot, moves by suppose 300 degree. Okay, suppose it moves at 300 degree. Now this particular movement of 300 degree is going to create a tension to the other end. It is going to create a tension to the next bond, which will also force the carbon, which will force this particular bond to move by a lot of degree, which will again force the bond downstream to it to move. So this entire forcing goes on. So if every atom does not reciprocate to that same force, then it is not going to move. For example, suppose you are standing in a line and there are 500 people in front of you, or suppose there are 30 people in front of you. So suppose you now want to push. So you are pushing someone who is just in front of you. That particular guy or that particular person is going to move when you push. So that particular force is again going to get exerted onto the second man from you or the person in front of your person, right?
Yes, sir.
Now that is going to get conveyed, but how many person are going to get affected by that particular push that you are giving?
So it depends on how many persons are connected to me.
You have 30 people in front of you.
Yes, sir.
So if you are pushing one person,
Hmm.
then he is going to go and push the person in front of him. Again, he is going to lean on the person in front of him. That man is again going to lean on the person in front of him. So how long do you think that particular chain is going to go? Only till 3 or 4 people.
Okay.
Only till 3 or 4 people, because the man you are pushing, he also has a particular balance, right?
Yes, sir.
So though he pushes the other man in front of him, that push is going to be lot less forceful. If you are exerting 100, suppose you are exerting a force of 100 unit, okay. So your person is front of you is getting affected by 100, but when he is falling onto the other person, what is the force that he is giving? He is giving around 70.
Okay,
Now
I
the
got
third
force.
person is yes, the third person is again going to give a exert a force of around 50. The fourth person is again going to exert a force of around 20. So you see, just after 3 or 4%, the force is getting lost, and that particular small force of 20 and 30 is not going to move the other person in front of him. Clear?
Yes, sir. Got
That's what happens in the crowd, right? In crowd, what happens? It is really difficult to push someone. Have you ever got up in a local train from
it.
Bidharnagar
Yes, sir.
station?
Yes, sir.
So you know how crowded it is, and is it possible for you to move in that crowded environment?
No, it's
No.
quite stagnant.
Yes, you behave as the way as the crowd behaves. If the crowd wants to get up, get down at a particular station, you move into the crowd, you get up, you get down. Again, from getting up also is the same. You mix with the crowd and you get up. It's really difficult. If you try to push or force person or push person in front of you, it is not going to help, right? Because there is a lot of crowded conditions. In cell also, that same crowding is maintained. The protein which is present in the cell, they do not float when we swim. We are single entity in a swimming pool, but that is not the case in case of our cytoplasm. In our cytoplasm, there are a lot of different factors. There are thousands of different proteins. There are thousands of different molecules. There are thousands of different ions. All are in contact with one another. There's a molecular crowded condition inside the cell. And that crowd prevents the protein to have any structure that they want. Understand? To maintain
Yes.
a proper structure, they have to fold properly. The protein folding is necessary. That is why, though the loops also have a tendency to move around or get any structure they want, generally they don't have that particular power, or they don't change that particular orientation. They maintain that particular angular structure. Okay, they maintain the phi-psi bond. So by measuring the phi-psi bond, we can predict if a particular protein is folded or not folded. Okay, and that will be one of the criteria to understand how, if we have modeled a protein properly or not. Because when we model a protein, they are going to give us 4 models. So how would we know whether that particular, which model is correct one? Generally, there are parameters to check, but to ensure that if model 1 is correct or model 2 is correct, sometimes according to the parameter, though they give in a particular order, the best is the model 1, second best is the model 2, like that. Sometimes we find out that the fourth model is the best model. Okay, so there are multiple things that we have to check, and this Ramachandran plot is one of the most important thing to check whether the protein that we have modeled is modeled properly. Okay, because if it is modeled properly, then all the psi angles are going to be inside the allowed regions, and if it is not modeled properly, then the psi angles are going to be all over the place. Now it is a different issue that even if we have a properly, does that mean that any model that have psi angles in an allowed region qualifies for a particular proper protein? No, that's not the issue always. Okay, there are multiple parameters to check, but this is one of the most important parameters. Clear?
Yes, sir.
Any question regarding this?
No, sir.
Okay, very good. Okay, so we are stopping here today. In the next class, we're going to take it up from here, and we're going to discuss the other issues, like we're going to discuss the Lewis-Thal's paradox, how the protein folds, and then we're going to move into the protein structure part.
So
Okay?
I have
Yeah.
a question
Yeah.
regarding the classes.
Yeah.
So how many days we are gonna have the classes?
There will be 4 classes.
Okay, sir. And sir, then we will be completing within a month, right?
Yes, we'll be competing in 2 weeks.
Okay, 2 weeks. One more question I have.
Yeah.
In the last class, you told me to install the PyMol.
Yes.
So actually I did, but I don't think it is completely installed. Like there is no such thing called install has been showing on my screen. Like it's only showing me files after downloading the thing.
After downloading, can you share your screen to me?
Yes, sir. Just a second. So is it my screen is
Yes,
visible?
yes.
Okay, sir. So this is the thing I'm getting, only files.
No, not that. What you have downloaded? Go to the
So,
website.
just a second.
Simply write in Pymol. I think you have not downloaded the Windows version.
Okay. I think I downloaded from here.
Yes, click on it. Yes, click on download now. Download the Windows version, the first option. Yes, done. Simply install this. Yes, you see that this is the software. Double click on it and you're going to install it. Yeah. You have Windows machine, so you have to install the Windows version. You install the Linux version.
Yeah, so I mistakenly did that. Okay, sir.
Okay.
So we are gonna have the same timing, or you're gonna
Yes,
see?
going to have the same timing. I'm going to update you the class schedule by today or tomorrow morning. Okay.
Okay, sir.
Sure.
Okay, sir. Thank you.
Yeah, of course.